Hello, I'm Radhika Pande, a PhD student in Dr. Das's lab. I'm currently working on understanding the epigenetic mechanisms during inflammation in colitis. Chronic inflammatory bowel disease, also known as IBD, such as Crohn's disease and colitis, is characterized by gastrointestinal disorders that lead to chronic and relapsing intestinal injury. According to CDC, in 2015, approximately 3 million Americans reported being diagnosed with IBD. Reasons for causing these are unknown, but several factors such as environmental factors, genetics, diet, and microbiome composition play important roles in disease development, and the pharmacological therapies are limited. Previous studies have shown that a protein called a nerve growth factor plays a crucial role in developing different types of inflammation. And we believe studying the expression of this nerve growth factor during the development of colitis can prove beneficial for managing this condition. Our aim is to study the epigenetic regulation leading to change in NGF expression. To give you an overview about epigenetics, it is the study of changes in gene expression that occurs without any changes in the DNA sequence. There are numerous proteins whose function is to regulate gene expression, and there are multiple types of epigenetic modifications. But most studied are histone modification and DNA methylation. My current study focuses on DNA methylation, but for that we need to understand what methylation is. When multiple C and G dinucleotides are present, they are called CpG island. The enzyme DNA methyltransferase adds a methyl group to cytosine. Previously, it was thought that methylation will always turn off the gene or make it transcriptionally inactive, but now our understanding of it is rapidly changing. Now the methylation or hypermethylation can turn on the genes to produce proteins. There is a protein called methyl CpG binding protein, that is MECP2, which binds to these CpG islands and helps regulate gene expression. Other transcription factors and RNA polymerase also bind to it for active transcription. For these experiments, we used a widely accepted animal model in which a chemical called TNBS is injected into rat's colon, which creates a robust inflammatory process mimicking colitis. As we wanted to study the role of methylation for the NGF gene expression during colitis, we used a chemical called azacitidine, which removes the methyl group, thus affecting the protein expression. We extracted DNA, RNA, and proteins from the colon for our experiments. Figure 1a is showing the colons from animals that have got different treatments. C shows the most inflammation as it is treated with TNBS, but in the D colon treated with TNBS and azacitidine, you can see the inflammation is reduced. Figure 1b shows the amount of methylation present on the NGF gene during different treatments. You can see that the amount of methylation is significantly elevated in TNBS treated rats and goes down in azacitidine treated rats. Graphs in figure 2 and 3 show a changes in the expression of nerve growth factor and MECP2 mRNA due to change in methylation during TNBS and azacitidine treatment. These qualitative and quantitative PCR mRNA of both NGF and MECP2 can be seen to be reduced significantly in animals treated with combined azacitidine and TNBS. The overall results indicate that azacitidine lowers the NGF gene methylation, ultimately reducing the overall production of NGF and MECP2 mRNA. Our protein data, not shown here, also shows the same pattern that azacitidine reduces overall inflammation in TNBS treated rats. So it can be concluded from these results that if we target specific proteins involved in epigenetic modulations, we will be able to control inflammation during colitis. I'm also interested in studying other epigenetic modifications like histone modifications and its crosstalk with DNA methylation for future directions. I firmly believe that Epigenetic modification can serve as a potential therapeutic options for several diseases. Thank you.